the theoretical basis of this experiment is on nerve muscle junction or neuromuscular junction. Okay, this is going to be an experiment on amphibian or frog or toad about the contraction of the muscle following a stimulation. This is called a muscle twitch. Okay, this is on nerve muscle physiology only. So it's a practical based on nerve muscle physiology, which we will not do, only visual representation of what is done actually in other laboratories. Okay, for the like calculation of different periods, resting period, like latent period, relaxation period, contraction period. So we are going to see it through the visual representation. What is the aim of the experiment? Simple muscle twitch. Now what is this muscle twitch? It is a, what is this muscle twitch? Already written here. When you give a single brief electric shock to a nerve of a skeletal muscle, it gives rise to a brief contraction of the muscle followed by relaxation. What does it mean? If you give a stimulation, a proper stimulation or adequate stimulation to the nerve supplying a muscle, then it will cause the contraction of the muscle followed by relaxation. Okay. So what are the materials required here? There would be a nerve muscle preparation, means a frog, then the entire preparation of the frog in a experimental setup. The nerve that is involved here is sciatic nerve, and the muscle which will be tested for this contraction or for this simple muscle twitch is gastrocnemius muscle. Can you tell me, anyone, what is the location of this gastrocnemius muscle? This one. This muscle is called the gastrocnemius muscle. There are two heads of gastrocnemius. This combined together to form the tendo-Achilles or Achilles tendon. Okay, and beneath that gastrocnemius muscle is the soleus or the peripheral heart. Now, what are the materials required here? The recording device, that is a kymograph. It would be a big drum or cylinder and then covered with a smoked paper. You will see in the subsequent slides. And there would be a fixed a tuning fork at 100 CPS for time tracing. You will get to know what is the use of this tuning fork and how it is calculated on the kymograph. See, this is the entire arrangement. This is the drum or the kymograph. Okay, and here the frog or the setup of the frog or amphibian will be placed here. This is one short handle. This is the long handle. This is the lever. This is our current source. We will be giving a current source. This current these two rods of electrodes will give stimulation to the nerve. Which nerve? Sciatic nerve. This nerve travels from the lower lumbar region to the back of the leg till the back of legs, back of thigh to foot. Okay, so this is the back nerve. This is the primitive model. Okay, similarly, what is this? This is the kymograph. This is the amphibian table, the frog table, where that frog would be placed. It would be cut, it would be dissected. Only this part would be used. Okay, a part of the spinal cord and a nerve coming out of this, and the muscle. Which muscle? Gastrocnemius muscle. Okay, let's see. Let's see. This is the frog board. Okay, where the frog would be placed. This is the muscle lever. This is the like short lever. This is the long lever. Okay, this is the myograph stand. Myo means muscle. The contraction of the muscle is estimated, so this is called a myograph stand. And this is the image, or this is that electrical stimulation would be given. The frequencies here, you can set the frequency from 1, 2, 3, or the voltage can be set. It would be repeated or single stimulus. We can set that also. This is the instrument used to give the stimulation. Okay. Now, how will you prepare that frog or toad? What you need to do? You can't do it on a live frog. What you need to do? पहले इसका इसको पैरालाइज कर दो. How to paralyze it? By stacking on it vertebral column. Bit of inhuman, but okay, you need to do this practical for this estimation of for this simple muscle twitch. That's why we don't do it now. There is no presence of here toad. This instrument is not here, but how it is done or how it was used to done. First of all, make the frog paralyzed by stacking its vertebral column or spinal cord. Then insert through this great foramen a pithing needle. This needle is inserted through the great foramen. Suppose it is inserted from here and it is put below along this entire spinal cord so that the spinal cord becomes damaged. उसका spinal cord को हम damage करेंगे. Similarly, the needle would be put into this frog's brain also. Its brain would also be damaged so that it does not means it's completely paralyzed. There is no other function of its nerves. Okay, only thus this part of the nerve is like we can preserve this nerve, sciatic nerve, along with that vertebral segments. Only this part is preserved. The rest of the spinal cord and brain is damaged. Getting this? The rest of the spinal cord, this is the spinal cord, no? It runs back here. The rest of the spinal cord is damaged, including the brain also, by this needle. Okay? So only that particular segment is preserved, along with the nerve coming out of that, and at the end of the nerve is the gastrocnemius muscle. Okay. See how it's done. This is our desired segments. Suppose this is the sciatic nerve. This is the thigh muscles. Okay. This is the gastrocnemius muscle. See, this is the gastrocnemius muscle. This is the tendo Achilles. This is our vertebral segments, which is necessary. See, only this part is necessary for the experiment. Getting this? Only this part is necessary for the experiment. The rest of the spinal cord and the brain are damaged. Okay, and also the other parts are cut. See, which is this muscle is gastrocnemius. This is the tendo Achilles or Achilles tendon. The muscles unite to form a tendon here. Okay, this is a hook. A hook along with a hook is used. Suppose some hooks are used here so that tendo Achilles is stabilized. And only this part is required for our experiment. Suppose this entire part is not required. This knee joint along with some part of the femur is preserved, and this knee joint will be put strongly in the table. इसको fix करेंगे. The knee joint would be fixed on the table. Again, the journey of the sciatic nerve from the sciatic nerve plexus, it's coming here at the back. I told you it's coming at the back. Okay, the back of thigh and going behind the knee joint. This is the tibia. Okay, this is behind the supplying the gastrocnemius muscle. So along with that muscle, with that nerve, these are carved out. Okay, this is the tendon. Now we are getting this. See. This is the spinal nerves combining and forming the sciatic nerve. Only this part is required for our experiment. Okay, gastrocnemius muscle. This may be the hamstring muscles. Okay, these are the hamstring muscles. Okay, after we have cut it out, now see, this part is the knee joint. This part is the sciatic nerve, and this is the spinal segments. Got it? Knee joint, gastrocnemius muscle, Achilles tendon, a nerve यहाँ से जा रहा है. Then this part should be in the thigh area, but it is carved out, and this is the spinal segments here. Okay, another representational photo.
I'll be continuing from here. So before starting the muscles twitching experiment, we need to have this solution. Okay. This is known as ringer lactate solution. Okay, so note down the composition. Okay, what is it comprises of, like NaCl, 0.6, potassium chloride, then calcium chloride, sodium bicarb, sodium phosphate. So this solution is the ringer lactate solution. So it has some specific functions. So it is given in your book. So you go through it. Okay. So like it maintains the morphology, then the conduction, okay, then the membrane preservation, okay, these things are there, okay. You go through this. So this is the picture, okay. Now this is the nerve, okay, the sciatic nerve, and this is the vertebral column, okay, and these are the electrodes which is connected in the nerve for stimulus, okay, and this is the gastrocnemius muscle, and this is the tendon which is connected to the short arm, okay. This is the short arm, with that side it is connected, and this is the long arm which will go to the drum, okay, and this is the knee joint, okay. So here the stimulus will be given, and then it will be contracting, and there in the drum the graph will be figured, okay. So it will be depending upon the stimulus. So the stimulus will be given to these electrodes, okay. So this is the vertebral segment, this is the nerve, this is the muscle, and this is the tendon. With this, with the short arm it is connected, and with the long arm, the picture will be, the figure will be drawn here, okay. So we will be controlling with this one, okay, with this machine, okay, the stimulus. So with each stimulus there will be contraction and relaxation. So this graph will be telling us where the point of contraction starts and from where it relaxes, okay. So components of simple muscle twitch. So there is one picture, it will be shown on the subsequent slides. So one point is latent point. So this, what is this point? The period from the point of stimulus to the point of onset of contraction. So this period depends upon few points, like the conduction of the impulse along the nerve, the neuromuscular junction, then excitation, excitational contraction coupling, stretching of series of elastic elements like tendons, and inertia of the recording lever. We will be showing this one on the after subsequent slides, okay. It will be coming. Then the second point is contraction period. So what is the contraction period? Period between onset of contraction and the point that corresponds to the peak of the contraction, okay. Wait, see this one, see this graph, see, this is the point of stimulus, okay. This is the latency period. So here the contraction didn't start, okay. I told you now there are certain components on which this latent period is depend. So after that the contraction starts from here till the peak, okay. This is the contraction period. See this CP, a midline is drawn from the peak to the base of the point of stimulus. So this line, so this is the peak muscle contraction, okay, PMC. Then from there it drops. So from here the relaxation period starts till the baseline, okay. This is the relaxation period, okay, or point of relaxation, okay. So there are certain frequencies of these waves through which we will be calculating it. So suppose if you consider this the middle line of a wave or the frequency, so till here it is 0.01 seconds, or the lower portion from here to here you can consider this also, 0.01 seconds. So per cycle it is 0.01 seconds, okay. So the contraction period I told, the relaxation period is, as I told, period from which the peak of contraction to the end of relaxation. So till end it is the relaxation period. The maximum height of the contraction I told, from the point of maximum height of contraction at the baseline of the tip of maximum twitch, okay. So this latent period depends upon these factors, okay, like conduction of impulse along the nerve, the neuromuscular junction, excitation, contraction, coupling, stretching of this series of elastic elements, and inertia of recording lever.
Let me simplify it. First of all, it is based on the two properties of muscle and nerve. Property of nerve is excitability. Upon giving a stimulation, the nerve will be excited. Means it would be carrying the stimulus to its end point. Okay, excitability and the contractility of the muscle. Muscle must be contracting. Okay, so see, we know here these are the electrodes. These are the electrodes. Here we can like fix the voltage, fix the amplitude, fix that like we can set it up. Frequency, amplitude, then voltage also, and okay, how many times repetition of the stimulus? Okay, once, twice. This is done for only for stimulation. This is the nerve. This is the vertebral column. Focus on 1 minute. Okay, it's not very tough. This is the vertebral column. The nerves coming out. The nerve is called sciatic nerve. This is the gastrocnemius muscle. It is fixed here. It is the knee joint. It is fixed here on the frog table. Now it is hooked here. Okay, it is hooked here and it is tightly attached to the short handle of the lever. Okay, now suppose I have given a stimulus here. What will happen? This stimulus will be carried through the sciatic nerve. It would cause a contraction of the muscle. This muscle contraction would pull it up. This would be an isometric or isotonic contraction? Isotonic contraction, because the length of the muscle will be changing. Okay, suppose this length is this. This should be changing like this. Okay, it should be shortened. On shortening, this would be pulled and this would cause a change in the, you have seen in that previous images, this is the kymograph. Okay, if this is pulled here, so this would change. Okay, this would also cause a, like this would also, like if it is pulled from behind, then this would be shifting like this. Okay, so upon shifting, why this graph forms like that? Graph forms like that, not this. Ek to hai cylindrical. Okay, and one thing, if it is, suppose this is the lever, it is connecting here and it is moving like this, so it would be causing like a, it would be making an arc here. But as the kymograph is moving, in a, it's rotating around itself. Okay, suppose the cylinder is there, its cylinder is rotating. So if that lever is changing its position, so this would cause a shape like that. Okay, why it's causing a shape like that? Because the kymograph is moving. Suppose this is the kymograph, this is the lever here. So if it moves, so it will form like that. Get it? This is continuously moving. This is continuously moving at a particular speed. I have set it up. The speed is suppose like 10 revolutions per minute, 20 revolutions per minute. So I have set it up at a particular pace. Now upon stimulation, it would cause this type of graph. Now sir has already told you about the latent period, contraction period and the relaxation period. Okay, latent period ka kuch factors hai. I think you have written it down. Okay, it would be asked in the exam also. And how this kymograph is made. This kymograph is cylinder. Around that cylinder, a white paper is wrapped. That white paper is like not stapled. It is like glued. Suppose a paper is there. A paper is glued to it, white paper, and now this paper would be smoked. This paper would be smoked. Suppose I light a candle here, and above that candle I make it, I place it here, and I make it like rotate like this. So what will happen? The smoke would be collecting here. It would be all smoky. That paper would be black now. You see the image. The kymograph is black in color. Black in color. Okay, it was smoked properly. That's why it's black in color. That tip of the lever actually has something sharp. Suppose a sharp plastic or sharp like something sharp that would cause a mark on that paper. That's why the sharp pointings at the end of the lever would make a mark like that. That means the smoky paper would be marked like that. This is moving. This is moving continuously. This is moving continuously. Upon giving stimulation here, muscle contracts. This is attached to here. Yeah, this is suppose here there would be a hooked would be there, and it would be contracting, it would be causing a change in the lever, and that change would be shown in the kymograph like that. More contraction, more height. Got it? Less contraction, less height. If it is contracting for more period, it will be traveling on a long distance. Okay, the area under the curve will be more. See, if it is pulling now, it would cause a change in that lever. Got it? Okay. At the end, it would form a graph like that. Okay. Here it is the latent period, contraction period. This is the maximum contraction. See, maximum contraction means height. If the height is like this, that means the contraction is less. If the height is more, there is more contraction. Okay. If it is like this, it is sustained contraction, means contraction for a longer period of time. And now another thing you may ask, like sir, what is this 0.01 second? Where is this graph coming from? Where is this graph coming from? Suppose you know tuning fork. Tuning fork dekha kabhi? If you didn't see tuning fork, Google it now and see how tuning fork looks. It has two handles, a base and a holding part. Okay. So we have taken a tuning fork whose standard vibration is 100 CPS. There will be 100 vibrations per minute. Okay. In 1 minute there will be 100 vibrations. And at the top of that tuning fork handle, suppose these are two handles now. At the top of one handle, I place something, and after striking that tuning fork against my hand or against something, I make it vibrate. The vibration is 100 per minute. Then I would place the tuning fork on that same paper, black paper. It will be moving. That's why, suppose it is vibrating at 100 per minute. So this one vibration will be corresponding to 1 by 100 or 0.01 second. Now get this. Before doing the experiment, मैंने क्या किया? Tuning fork के ऊपर एक sharp चीज लगाया. Suppose I know this tuning fork is going to vibrate at 100 per minute. मैंने इसको strike किया. It is vibrating at 100 per minute. The kymograph is moving fast. मैंने इसको place किया and it's making a graph like that. Okay? बोलो. Sorry, 100 per second. 100 per second. It would be 100 per second. And okay, after placing that tuning fork now on the kymograph paper, it is making a graph like this. Okay, and this the frequency is 0.01 second. Sorry, the time period between these two two sloughs or two amplitudes is 0.01 second. So the entire vibration takes, like in 1 second there would be 100 vibrations. Okay, so that.
Calculate the time.
Okay, only for this only, to calculate the time period. If I ask, so what is the time period between this latent period? So this point of stimulation, the point of contraction, say there is one complete wave. That means 0.01 second. Similarly, what is the period of contraction here? This contraction period, calculate 0.1 second, 0.1 second. Then this would make be 0.03 second, not 0.1 second, 0.01 second. This is 0.01 second, 0.01 second, and this would make one wave, means 0.01 plus 0.01 plus 0.01, that means 0.03 second. Similarly, tell me what is the relaxation period here? 0.04. Getting this?
Okay, this is for the simple muscle twitch.
इसमें क्या है? Or whenever you are going to write it, you can write it now. Theory लिखोगे, theory part थोड़ा सा. Start muscle preparation, जो भी देखा ना, कैसे prepare कर सकते. And like not much, only in brief. And set up थोड़ा बहुत. If the photo is available, one photo, one image, give an observation table, does it? Okay. And then go there, and then reach there, and then causing this change in the lever. Okay. And also there is some viscosity in the muscle. and there are other properties. I think you have written this. Contraction में there is some delay. Neuromuscular junction में थोड़ा delay होता है ना? Traveling of that stimulus that from pre-synaptic to the post-synaptic neurons. The delay is causing that latent period. Okay. Now this is the this is contractions. Okay. Now these are after the contraction. There is some subsequent some vibrations. These are not required. The muscle is contracted and relaxed till here. Okay. What is this? Every peak and slough is how much second? 0.01 second. Why? Because the frequency of tuning fork is 100 per second. Thank you.